Got to the workout room. Shelby had already been there and had left. And I saw out here in the breakfast thing, the bowl of nuts that are healthy for you was almost empty. And the cranberries were almost gone. And there were no raspberries left on the fruit tray. So already internalizing those nutrition things. Though I also noticed that quite a few of the chocolate cakes were missing from the buffet last night. I'm not sure how that fits in, but... oh, there you go. I like that. We have a very exciting morning lined up for you, I think. And maybe it's just because I'm a bit of a research geek that I like this kind of thing. So the first presentation is by John Sondek, who is came to to a meeting that we had of scientists in I believe Miami, and it was the first science meeting after the Cure-OM program was started. And he did a presentation about G proteins. So you've heard GNAQ, GNA11 as being drivers in ocular melanoma. And I understood the letter G, and that was about as far as I got. But if I gather right, the concept is that doing something with the G protein itself is extremely challenging. But the G protein, there are some downstream things that you can kind of do some interdictions around, or potentially do some interdictions around. It's a kind of a novel approach. And all of the people, after that science meeting, all the people in the room were buzzing about this concept and the idea that you could do something more about this. And then as Cure-OM developed and matured, we were able to secure funding through the generosity of some donors for a peer-reviewed grant process. And so we did an RFP and asked people to write grants. An RFP is a request for proposals. And we requested proposals for securing grant funding. And we got a number of grants in. We went through a rigorous review program from top scientists. And there were two different grants. I don't know if you even know this or not. There were two different grants. I thought I told you this. They kind of bubbled up to the top as being of the greatest interest. And the scientific review group said, we can't decide which one you should fund. You have to make that decision. And one of them had a high likelihood of being successful. So there's proven track record. It's pretty safe. We kind of get that it's going to, if you do this research, it's really going to work and it'll be fine. The impact on ocular melanoma would be, it'd be there, but it wouldn't be kind of groundbreaking. The other one, which was Dr. Sondek's approach, it was not as clear if the ultimate ends could be met because it was paving new ground. It was kind of carving out new territory. So it's a little bit riskier, but the impact, if it worked, the impact would be really big in understanding ways to attack this cancer. And so we actually went back to the donor who had made the grant possible and said, here's what we have. There's one that's high risk, high reward, one that's lower risk and lower reward. Which way do you want to go? And they said, I want to fund the high risk, high reward one. And I think this morning we're going to hear at least some of the results of that grant funding so far. So Dr. Sondek, to you.
Tim, thank you for that wonderful introduction. First, let me say that it's an incredible honor to be here. I don't normally get to speak to anything but what you call yourself a geek scientist or something. That's my venue. So it's a pleasure to be able to work with and see how perhaps some of the work that we're doing can directly impact people's lives. What I want to do is talk about this protein here, G-alpha-Q. And the reason I want to talk about this protein is because it is the major driver for uveal melanoma. And the reason I can say that is because of some giants in the field that came before I even knew what uveal melanoma or ocular melanoma was. That is, these two papers, one by Bill Harbour, who's intimately associated with this group, and Boris Bastian's group, realized that this protein, the lingo here is GNAQ, is the gene, and that gene is transcribed and translated into a protein which is called G-alpha Q. So one's the gene and the other's the protein, and I will typically be talking about G-alpha Q, the protein. They realized that in about 50% of all uveal melanomas, this protein is mutated. And then similar work came out shortly thereafter saying G-alpha 11 is mutated in another 30% of these cancers. So approximately 80 to 90% of uveal melanomas have a mutation in G-alpha Q or G-alpha 11. G-alpha 11 is very similar to G-alpha Q. So I'm just going to lump them all together and say G-alpha Q, because the mutations in both of these proteins are essentially identical. The downstream signaling that I'll be talking about is essentially identical. So that's the protein that we need to focus on, because I will describe to you how the activation of that protein causes the proliferative response that leads to this disease. It's one of the major contributors, and I'm not trying to oversimplify this. There are other mutations that cause other aspects for this disease, but clearly this one is very important. If you knock down this gene in, for example, a uveal melanoma cell line, those cells will die because they're oncogene addicted. And so that's the concept that we're going to try and address here today, is how to stop signaling by this protein. And in order to talk about the signaling, I just want to give you a little back history. So you probably all read in the newspaper at some point in 2012, a Nobel Prize was given in chemistry to these two very nice-looking men over here. One is Bob Lefkowitz. The other one is Brian Kabilka. And they received this prize for doing research on these proteins here. So this is a G protein coupled receptor. In the lingo of the field, is called a GPCR. These proteins, and there's about 700 different ones of them in humans, their role in life is to move information from the outside of the cell that's demarcated by this membrane to the inside of the cell. And they do that by binding compounds on the outside. This whole structure changes its shape. That change in shape is then is sensed by a protein on the inside called a heterotrimeric G protein. It's called a heterotrimeric G protein because there's an alpha subunit, such as G-alpha Q, and a beta-gamma dimer that doesn't come apart. That information is transduced across. So, for example, the drug never goes across the membrane. Just the information is transduced through the conformational change in the receptor to the trimer on the inside. That trimer on the inside then becomes activated. The way it becomes activated is it kicks off this little molecule called GDP. It stands for guanosine diphosphate, but you don't need to know that. And then it loads another small molecule that's very similar called GTP. The binding of GTP causes this portion to break apart. Those two portions of the trimer then can go down and bind other proteins and cause them to do things in the cell. I entered this field sometime around 1992, and I had the joy of being part of the team that did some of the first structures of heterotrimeric G proteins with Paul Sigler at Yale. A Nobel Prize was given in 1994 to Al Gilman and Marty Rodbell for discovering these proteins. So this is a very conserved, very important signal transduction cascade in humans that moves all sorts of information from the outside of the cell to the inside of the cell, whether it's sight, whether it's taste, whether it's a hormone, a neurotransmitter, etc. And in this particular disease, the mutations that hit G-alpha Q cause G-alpha Q to always be in the on position. And so the basis of this research is, can we stop G-alpha Q from signaling to its downstream partners? That's it. That's the entire premise. So in a very simplified fashion, this is what we're trying to do. So you have the GPCR activating the trimer. Here's the GDP. It catalyzes the release of that GDP and the loading of GTP such that it's on. The trimer splits apart, and each piece of that trimer can then interact with downstream effectors. Here are the mutational frequencies in G-alpha Q. So you can go to this website, and you can find all the tumors where it's been looked at. They're almost all uveal melanomas, almost all of them. And those mutations are at 2 sites, arginine 183 and glutamine 209. These are catalytic sites, residues within the protein that are required to hydrolyze that GTP to GDP and shut it off. So when you mutate these 2 sites, which is almost all of them, by the way, something like 95% are at these 2 sites, that alpha subunit can then not hydrolyze the GTP back down to GDP and shut it off. So it'll always be on. And that's just shown here because when you mutate here, you're going to cut off this leg of the signal. You're going to be stuck here. You're always going to be going in this direction. You're going to interact with your effectors, and you're going to be leading to proliferation. So the idea is let's stop it right here. Now, one thing that I should emphasize is that I realized the other day that we have all these clinical trials going on. But those clinical trials are essentially taking drugs that are known for other cancers and repurposing them to uveal melanoma. Nobody is looking here. This is essentially one of the key steps to this process, and you will not find a pharmaceutical company that will study this process, and I've had it said to my face because it's essentially an orphan disease. And I'm exceptionally grateful for this foundation for allowing us to move forward with this process. All right, so here's a simplified diagram, somewhat simplified anyway, from what you saw the other day. And I just want to emphasize that here's uveal melanoma over here. 80 to 90% is driven by G-alpha Q. It goes through one of these 2 effectors. We don't know which component of these 2 effectors is responsible for this disease state. It may be both, it may be one or the other. I'm interested, and I have active research in both of these areas. But nonetheless, you funnel down this way till you get to something called GNAQ, which we heard about the other day, and to ERK, and then into the nucleus where you drive gene expression and proliferation. This is the much more common area, and this is where you hear things like the MEK inhibitor that was repurposed from this side to over here. So in this case, cutaneous melanoma, there's about 50 times more patients that have cutaneous melanoma relative to uveal melanoma. And there are very good inhibitors for some of these processes. Very famous MEK inhibitors, very famous BRAF inhibitors. This pathway over here is driven by mutations in NRAS. I won't really get into this, but this is also related to the G protein here evolutionarily, way back in the distance. But NRAS is mutated, BRAF is mutated, and you drive in through this process here. However, we're over here, and we're activating G-alpha Q. We're driving down this way. So this is an entirely different pathway for this particular mutation. So some of the, I personally believe some of the particular compounds that are being used in combination would not be nearly as efficacious as if you had a G-alpha Q inhibitor that you could then use in combination with some of these drugs. Okay, and just for a point of interest, you can get these mutations in your skin, but they drive not only a proliferative process, but also a differentiation process, and they end up being a blue nevus. So they don't typically cause cancers, although they can. In cutaneous cells, they'll typically drive this melanoma. For reasons we don't understand, in the eye, that process of differentiation is absent, and you simply get the proliferative response leads to the tumor. All right, so a couple of years ago, we did this structure over here. This is G-alpha Q in its activated state bound to one of these effectors, phospholipase C beta. And a few years back, another one of these structures had been done by another group headed up by John Tessmer of P63 ROGAF bound to G-alpha Q. And what we realized is that these 2 proteins are interdicting this. Their G-alpha Q is binding exactly to the same to both of these effectors. So the idea is if we can block the major interaction here, you would stop G-alpha Q from interacting with PLC beta, stop G-alpha Q from interacting with P63 ROGAF, and then you would stop this downstream signaling from both of these. So that's the concept that we wanted to put forward. And I put a movie in here just to emphasize all this stuff, okay? Now, what I want to emphasize is these are not cartoons in the sense. I spend my whole life making these things from atomic information. So we're at the level of atoms here. So here's the protein, here's the alpha subunit, here's the GDP. Now this is going a little bit fast. These are the regions that are called switch regions that change when you have GDP bound versus GTP. And here's another thing. So those are the regions that move and allow it to interact with the effectors. So this is just to emphasize that that is actually electron density indication of where the atoms are in space. So that's what we're spending our whole time. Let me stop right here for a second. We spend our entire time, well, not my entire time, but a lot of it, looking at these little wire mesh things because they are an indication of the atoms in space, which we define by something called X-ray crystallography. And then we make these pretty pictures from that. But we know where all these atoms are. Those two things in yellow are those two residues that are mutated in that chart that I showed you of arginine 183 and glutamine 209. They are required to hydrolyze that small nucleotide, GDP, in the background there. So when you mutate those two sites, you can't turn it off. And that's how this works at the molecular level. Okay? All right, so that's the alpha subunit. And now I just want to emphasize, okay, so here comes the PLC, and they lock in together. And we have structures for all of these compounds. And we take years to make these structures because they're very difficult. You actually have to make crystals that are smaller than a grain of salt from your salt shaker. And then you put them in an X-ray beam, and then you have to work from there. So it's just going to march through the different domains in the PLC, and a domain is simply a small piece of the entire protein, usually something that will fold in and of itself. But this is just a complex of two proteins, the alpha subunit and the PLC, the effector complex. This is essentially telling us how G-alpha-Q activates this PLC molecule. Now here's the last domain in the PLC. It's called a C2 domain. You obviously don't need to know that. So this is sort of evolutionarily a compact structure that there's a whole bunch of these PLCs, and only the PLC betas interact with G-alpha-Q because of this little piece here, which is essentially a helix-turn helix, or I'll refer to it as a peptide. But it's the basic piece that binds to G-alpha-Q. Here's the surface of G-alpha-Q, and I just highlighted the switch regions, those pieces that move based on what nucleotide there is. So you can see G-alpha-Q interacts with mostly this piece of PLC beta through this little cleft here. And the idea is we want to find molecules that bind in that cleft, prevent G-alpha-Q from interacting with the PLC, and then you can prevent the activation of its downstream signaling components. It's that's the basic concept. All right. So how are we going to do that? What we're going to do is we're going to take that peptide, we're going to put a fluorophore on it. It's a thing that interacts with light. And we're going to take polarized light. This is linear polarized light, just like your sunglasses would be. And that linear polarized light, if it hits this peptide, which corresponds to that piece that I showed you in the movie, with the fluorophore, this thing is tumbling rapidly in space, and the linear polarized light, it drops off rapidly. However, if it's bound to G-alpha-Q, that is very, very big relative to the peptide. It will not tumble as rapidly, and you will not lose that polarization signal as quickly. And you can measure that through sophisticated means. So that's what we did. So if you take just the peptide from PLC beta 3 and add it to a cuvette, well, you have it in a cuvette, and you add in more and more G-alpha-Q, you get this, what's called a binding isotherm, and from it you can measure an affinity. And if you only have half of this thing, half of this peptide, you cannot bind G-alpha-Q, so you don't get any change in signal. The fluorescence polarization doesn't go up. And then we can make mutations that increase the affinity of the peptide for G-alpha-Q so that the fluorescence goes up faster, the polarization goes up faster with less G-alpha-Q. So this would be a high affinity peptide binds to G-alpha-Q. Now, we have a way of monitoring activated G-alpha-Q in what's essentially a test tube. So if you take activated G-alpha-Q, it binds to the peptide very nicely. Inactivated G-alpha-Q, no binding, no change in fluorescence polarization. Now, importantly, there is a bunch of other G-alpha subunits in your cells that are important, and this peptide does not interact with any of those other G-alpha subunits. So this is a specific readout for G-alpha-Q that we're going to use in an assay to look for inhibitors. And the idea is shown here, where in this case, we'll just use full-length PLC beta. And if you take full-length PLC beta, you can displace that peptide. And then if you displace the peptide, the fluorescence polarization will again drop. This is basically a control, but the idea is at some point we're going to replace the full-length PLC beta with a small molecule compound. And that's exactly what happens if you take full-length PLC beta in a system that has this already complexed with it. The polarization will drop off as a function of the full-length PLC beta 3 concentration. If you have a mutant form of PLC beta 3 that does not bind G-alpha-Q, you cannot displace that peptide. So this would be the equivalent of a small molecule that doesn't do anything to G-alpha-Q, and this would be the equivalent of, say, a small molecule that displaces PLC beta. Okay, so now we're all set up to run a high-throughput screen. And here's some actual real data that I simplified for this talk, which is essentially if you have inactive G-alpha-Q in a sample with this fluorescent peptide, your fluorescence polarization is down here, as I just walked you through. If you have activated G-alpha-Q in this sample with your fluorescent peptide, the polarization is up here. Now, if you have compounds that interfere with G-alpha-Q's interaction with the fluorescent peptide, you'll drop the fluorescence polarization, and you can read this out. Now, what we want to do is, and what we need to do in sort of high-throughput screening, is to look at this for lots and lots of different compounds. So we're going to be using it to look through libraries of hundreds of thousands of compounds. And so you have to automate this into a high-throughput fashion. And that's what I've sort of illustrated here. You run these assays in these plates. They're called microtiter plates. You can have them in 96, 384, 1536. So this is essentially 100 different wells that you can look at individual compounds in, maybe 300 wells or so here, and over 1,500 wells here. And then we're going to use robots to move these compounds around, move the G-alpha-Q around, move the peptide around, such that we can then take this plate, use it in another machine, and read out this fluorescence polarization. And we will look for those compounds that interfere or shut down the fluorescence polarization, interfere with the capacity of G-alpha-Q to interact with this peptide. And some of the data is shown here. This is simply a plate. Each one of these squares represents a well in the plate. These would be controls over here, controls over here. And here you have G-alpha-Q and your fluorescent peptide. And in most of the cases, the polarization is high. Now, each one of these will also have a compound in it. And all these compounds do nothing until you get to this one here. So this would be what you would call a primary hit. It, for some reason, causes the fluorescence polarization to drop. And there are a whole host of reasons this could be artifactual. But if it is a true hit, then it would be interfering with the peptide's capacity to interact with G-alpha-Q. And it would be a potential lead to stop this process in cells. And you then take this sort of compound here and you run what is called a dose-response curve. So here you would have decreasing amounts of that compound in these wells. And then each one of these would be one of those primary hits. And you can get from this a dose-response curve. These are all just controls over here. So you can get a feel for how much of that compound is required to prevent so much G-alpha-Q from interacting with this peptide. And it's a measure of the affinity of that compound for G-alpha-Q, if everything is working as we predicted. So that's what we're doing. That's why we're doing it. And this is where we are today. So I was given money by this esteemed organization to do what's over here, essentially, where we've taken 100,000 compounds that are sitting at UNC Chapel Hill. We've done this primary screen. You get about 62 of these primary hits. You then do these dose response curves, and you're down to about 5 hits. This is a hit rate of 0.005%. So this is really a numbers game. Now, I fully expect most of these may not go, we may throw most of these out for one reason or another, looking for that lead compound that we can then build up through something called quantitative structure-activity relationships, or essentially modifying that molecule to find ones that increase the affinity for G-alpha-Q from our original hit. And those would essentially be lead compounds for a drug discovery optimization. However, we've gotten much more efficient at this than we thought, and we're actually doing 2 other libraries. We just finished this one here of 17,000 compounds. We got another 12 coming out of this one. And we're doing another 100,000 compounds, and we're right in the middle of this one. So this is where we are to date. And over the next year or so, what we'll be doing is taking these compounds, figuring out which ones are the most reasonable in terms of chemistry that we can modify, in terms of their mechanism of action, in terms of how they interact with the proteins, to start modifying for lead optimization, for potential initially interpretation of signaling downstream, and then as leads for drugs. Okay, so 3 avenues forward. I talked to you about this one here. But we're also doing 2 other programs at the same time. And they all spin out of this work. One thing you can think of is, well, you can take that peptide, and you can use that peptide to inhibit G-alpha-Q. And that's true. That works perfectly fine. I could show you data where we've taken this peptide, we've injected it into brain slices of mice, and it shuts down G-alpha-Q signaling like nobody's business. We've actually screened for even higher affinity peptides that bind G-alpha-Q and found them. What we're going to do with those is they're going to be the leads for what's essentially synthetic peptides. You can make a peptide. You don't have to genetically encode it. You can make a peptide using what's called a peptide synthesizer, and you can modify it in ways that increase its bioavailability, increase its stability, and so on. And you can then use those to inhibit these signaling cascades. This is another distinct program that we're working on right now. And then finally, you could imagine, for example, encoding these peptides in adeno-associated virus, for example, and using them to interdict signaling cascades. I see these as 3 distinct but absolutely viable programs forward for treating this disease. And we're working on all 3 of these programs. Now, I just want to give you 2 slides because I said before that this is, I've had it said to my face that this is an orphan disease and nobody's going to work on it. And that's just the state of the world right now. But with this money, it's helping us to do essentially a workflow that's related to a whole host of cancers. So in many ways, we can be the drivers for the rest of the field. So G-alpha-S is frequently mutated at catalytic residues. If you take all the tumors where G-alpha-S, which is another protein related to G-alpha-Q but distinct, and look at its frequency of mutations, it's mutated in about 4% of all cancers. Again, at 2 places. These are called hotspots, by the way. So whenever you get a mutation frequency like this where it's always at the same place, it's called a hotspot. And whenever you get a protein like this that's found in a lot of different cancers where it drives the cancer process, it's called a driver, as opposed to, for example, a passenger mutation. So these are driver mutations for a host of different cancers. Now, just to remind you, if you put G-alpha-Q up here, these 2 sites are essentially, they are functionally the identical sites to these 2 residues here. They're just in a different protein, and they're flipped in terms of their frequency. They do exactly the same thing. They prevent the G-alpha-S from hydrolyzing that nucleotide. That G-alpha-S is always on, and it drives a proliferative response. And it does so in a host of different cancers. And here's the frequency. The guys in red down here, the tumors in red, are the ones that kill the most, are in the top 10 cancer killers in the United States. So we're talking about, like, for example, 15% of all pancreas tumors will have a mutation in G-alpha-S. The workflow that I described for what we're doing with the Cure Ocular Melanoma Foundation, which I'm so grateful for, can also be applied to these different tumors because of this process. Okay, now, perhaps the most important slide. This is represents a long-standing collaboration with Ken Hardin at UNC Chapel Hill. Gary Waldo did the structure of G-alpha-Q bound to PLC beta with technical help from Matt Barrett. Tom Charpentier started the entire program using peptides to inhibit G-alpha-Q. That's now been taken over by Stuart Endo-Streeter. We collaborate at UNC Chapel Hill with Bill Jansen and Melissa Porter in what is called the Center for Integrative Chemical Biology and Drug Discovery. Very long-term name. We have a whole host of other collaborators for synthetic peptides, which are really just mentioned at the end, some cancer biology, etc. Down here. Nano delivery, so some of these peptides can be delivered by nanoparticles, which is a whole other area of science. And then, as I said, I'm immensely grateful for Cure Ocular Melanoma for funding this work. I have to emphasize that I do not think I would be doing this work because without these funds, I may have gotten funding for it eventually, but it would have been a tough sell, I think. And it's immensely gratifying to me on a personal level to be associated with this organization and the things that they're doing. We also get funding from the National Institutes of Health, the American Cancer Society, and I'm forced to disclose that I have a small biotech firm that I just started, which is only peripherally related to any of the material I showed here today, but just to be on the safe side, I'm disclosing that. And then finally, I would just like to thank you for the opportunity to be here and give this presentation. Thank you.
17,000 compounds, looking for a handful of things that could become drugs that will have an impact on this cancer. It's so exciting. I mean, to me, this is astounding and really wonderful that you're able to do this work. So thank you, and keep it going. We're going to have a research Q&A panel, but before we do that, I wonder if there are any questions specifically about this presentation that people would like to address.
Sure.
Where's the link between your results and the drug company? Right? What's the next step once you synthesize what you want to do?
So once we get a compound that we're confident inhibits G-alpha-Q, the first thing we will do is we'll take a panel of uveal melanoma cell lines that have been genotyped for this mutation versus a panel of, say, uveal melanoma cell lines that do not have this particular mutation versus, for example, a set of cutaneous melanoma cell lines. And so the first thing you do is you squirt the compound on there, and what you would expect to see is that uveal melanoma cell lines that are addicted to G-alpha-Q will die. That's predicted because we can genetically ablate G-alpha-Q and show that they would die. So we're expecting those cells to die. And for example, cutaneous melanoma, they shouldn't have as dramatic an effect. Now, I mentioned this the other day to somebody, and I said, what I'd really like to have is, for the next step after that, you want to go into what's called a mouse model of uveal melanoma. Now, there is really no good mouse model of uveal melanoma at this point in time. I know of one person that has a mouse model, but it's not optimized. And so what you do then is then you take the drug and you put it in a model of an organism such as a mouse where they have been genetically modified to recapitulate as best as possible this disease, presumably by putting, say, constitutively active G-alpha-Q under an inducible promoter that expressed only in the eye, and you get melanoma in the eye, and then you treat the mouse and you see, does it have an effect? So that's sort of the basic research pipeline that people follow. And then after that, then you're talking about, well, can you use it in some sort of IND clinical-based program with clinicians?
So a little bit of definition here.
Sorry.
That's okay. This the drugs that we're so excited about that are taking the market in cutaneous melanoma right now, the ipilimumab and the PD-1s and things like that, all started with this. And so you start in the lab working with these analyses and these studies and doing these high throughput screenings and those kinds of things. And then it moves to cell lines and to animal models and then into early clinical trials. IND is an investigatory new drug, and it's a specific licensing procedure through the FDA that says, okay, we're going to start looking at this, and it requires all these approvals. The whole thing is somewhat slow and ponderous, but a lot of people are giving a lot of thought to how to make it faster. And I'm happy to say that the person in charge of oncology drug development at FDA, I think, is being pretty creative around some of the regulatory issues. How can we move this along a little bit better? And so one of the drugs that was approved a couple of years ago, vimorapinib, had a very short timeline relative to what other drugs have taken. So we're trying to make all of this faster. The key really is the ability to get the research done. And as I'll talk about later, that's really tied to funding. So as you said, if we hadn't had some donors who stepped up and said, I want to fund research, this could have sat on the shelf for another 10 years, or maybe forever. And that's the key part of it. And then the other key part of it is around getting people into clinical trials, and we'll be talking about that a little bit later on as well. It's just critical. Any other final questions for John before we move to the panel?
Since it is so rare, then what, I ask myself anyway, what is it about all the others that, that makes it so rare? Why is it that, for instance, I have in my family melanoma, skin melanoma, but what makes my melanoma then so much rarer?
So
what do they have that prevents them from?
Yeah, so for people who couldn't hear that well, it's a great question. So melanoma in general is a relatively rare cancer compared to breast cancer, lung cancer, or colorectal cancer. Ocular melanoma is only 5% of all melanomas, so it's even more rare. So the question is, why is it so uncommon? Why is ocular melanoma so uncommon relevant to some other cancers?
So I think there may be 2 points that I can make related to this. And one is the other day, and I attribute it to Bertel because he reminded me of this. First off, so the space of the size of the eye is much, much smaller relative to the skin, which is actually the largest organ in the body. So based on just surface area aspects, you would expect much fewer melanomas. And I would also say that you'd probably see a lot more cutaneous melanomas that are driven by this genetic lesion if you didn't get these differentiation process into blue nevi. Blue nevi is actually quite common. So I think the question was the flip side, why is the uveal melanoma so much more rare than cutaneous melanoma?
The reason I'm asking that as well is I have multicystic disease, and so I have a tendency towards, I was born with many moles, completely all over the scalp, back, everywhere. So it seemed to be, to me, I've had other cancers as well. So it seemed to me that there's some kind of a connection between multicystic and getting the melanoma. Also of the eye, because I have a family member that had the step before.
Can I take a stab at this as the one non-researcher, non-doctor on the group here? And while I'm doing this, I will ask Tara McCannell and Uveal and John, if you would just move up to the table here, perhaps, and we'll get ready for the research Q&A. So think about why does cancer happen, right? So we all have cells that get screwed up, and when cells replicate, sometimes things go wrong. It happens to all of us all of the time. And cells have design mechanisms inside of them that if they screw up, there are ways of fixing the screw-ups. They have a self-repair mechanism. And then they have mechanisms that if the self-repair mechanism doesn't work, they self-destruct. They're not supposed to live. And if they don't self-destruct, the immune system is designed to find those weird cells and get rid of them. So for cancer to happen, if we actually diagnose cancer, all of that has to fall apart. Right? You have to have a mutation that can't be fixed, that doesn't lead to a self-destruction, and the immune system can't find. So that's when the cancer actually happens. So the more of that problem that occurs, the more errors that occur in the cell, the more likely it is that somehow or another something will escape the mechanism. And that's why I heard recently that what happens to me is 60% of all cancer can be tied to diet and exercise. Right? So if you think about colorectal cancer being very common, colorectal cancer happens in the colon, which is exposed to all the things that we eat. And all the things that we eat impact those cells in the colon and do bad things to them. And so you can think about what happens in the eye, though. The eye doesn't have that kind of exposure to these mutating types of agent at the same level as some other organs have. Now, I'm not a doctor. I may have really screwed that up, so feel free to correct me. But it seems to me that may be a factor in this, as well as some of the kind of basic underlying biochemical factors. Okay, this is a great opportunity. Anything you want to know about what's happening in ocular melanoma research, you have an expert here who can address that at some level. All the way across basic science, people who are doing clinical trial research, people who are doing clinical care. It's really a great panel. And I have about 70,000 questions I could ask them, but I'm not going to ask any of them if you guys ask them questions. So, Molly, if you'll handle the microphone, and we'll move this along.
Dr. D'Amato, I've noticed that there's a fair number of newly diagnosed patients here. And having you involved in our life, I'd like you to share your experience in England in terms of fine needle aspirations, biopsies, second-generation testing, and in particular, the fact that you have actually done several times fine needle aspirations after radiation, because that really is not discussed much over here.
You've got, as we all know, three kinds of melanoma in the eye. There's the good, the bad, and the ugly. The good ones never metastasize. The bad ones nearly always do. And what I call the ugly ones are the ones where things don't quite fit. Genetically it looks bad, but histologically under the microscope it looks good. Or under the microscope it looks terrible with lots of mitosis, lots of cells multiplying, and yet it's class 1 where things don't fit. And there's a few outliers like that. And it's really important. They all look the same, by the way. When you look at the back of the eye, they look very similar. And the only way to distinguish what kind of disease you're dealing with is to do a biopsy. And that biopsy and that information is of tremendous importance to the patient. If they know they've got a good melanoma, then most of them feel much better than if they have been told they've got a bad melanoma. Not necessarily. There are some. We found out that just saying to a patient you've got a good melanoma isn't enough, because the patients don't believe you. And it's a real pity, it's a real shame when a patient's got an excellent prognosis for survival and they're miserable because they haven't believed you. So there's a certain technique in informing patients of the prognosis in such a way that they have confidence in the result. You've got to show evidence of, you've got to justify the evidence and convince them that you're being honest with them and you've got the data that means something. Conversely, in patients who've got a bad prognosis, there are lots of doctors who are dead against giving patients a bad prognosis if there isn't a treatment. I've had to stick up for myself and for my colleagues at lots of conferences, and I'm sure Tara's had the same experience. But as you know, patients want to know their prognosis, good or bad, and our psychological studies have shown that a bad prognosis is less stressful than no prognosis at all. Uncertainty is the most stressful situation because patients are not empowered to do anything. They're lost. Whereas if they know they've got a bad prognosis, they can find out what to do. They can find the experts around the world to consult, and they can do all sorts of things to help themselves. And it also influences screening, especially in a place like England, where you can't. It's a socialized medicine. The government pays for all the tests, but the government can't afford MRIs every 6 months on every single patient with uveal melanoma. But by doing biopsies and finding out which patients are going to have got a high risk of metastasis, we have been able to offer these patients 6 monthly MRIs indefinitely, free of charge, without insurance or anything, but only because of the genetic testing that we've done. Now, intuitively, you would imagine that first you do the biopsy, and then you do the radiotherapy. First you ask the question, and then you shoot afterwards. It doesn't make sense to shoot first and ask questions afterwards. But when we started off doing the biopsies first, there were a few patients who got a vitreous hemorrhage, and then when we came to putting in the markers for the proton beam or putting on a plaque, it was very difficult to do that because the blood got in the way. We couldn't see where the tumor was. It was really difficult to treat the tumor because the biopsy had caused the hemorrhage. And secondly, there are patients who are concerned about the risk of scattering tumor. Now, even if that risk is very, very small, especially if a lot of these patients, sadly, are going to get tumors elsewhere, we don't want any of them to blame that on the biopsy. We know that it's not the biopsy that has caused the metastasis because these tumors, they spread biologically, sort of, not physically, etc. But even if a small proportion of patients say that to themselves or their relatives feel that if they hadn't had a biopsy, they wouldn't have the liver tumor. It's false logic, but it's a powerful logic. And if it's going to hurt patients, then one way to get around that is to do the biopsy after the radiotherapy once the tumor is sterile. Now, it's been said that the radiotherapy is going to cause lots of mutations, and these mutations are going to scupper the genetic testing. But the chances of in order to detect a mutation, you've got to have a lot of cells having the same mutation. And the chances of the radiation causing the same mutation throughout the tumor are extremely small. It's going to cause lots of nonsense mutations and very few chromosome 3 mutations that really, really matter. So for many years, we've been doing the biopsies, more than 5 years, we've been doing the biopsies after the radiotherapy. They have the radiotherapy on the last day, on the way home, they stop in the hospital, get the biopsy, go home. No worries about spread, no worries about hemorrhage, no problems at all. And that's worked very well for us for several years. Do you have any comments, Tom?
No, I think that that last point about experience with doing biopsies after the tumor has been treated. We talked about this in one of our group sessions yesterday, and that could be something very valuable for people who had their tumors treated before the biopsy era. And I had a question for you. How long after, you said the last day of radiation, do you have experience with sort of several years after radiation?
We do have patients where genetic testing has been done years after the radiotherapy. And these are patients who've had a recurrence, have had the eye removed, and have harvested some of that tissue, and it has shown chromosome 3 deletion in those patients and so on, or not. If there's bulky tumor months after the radiotherapy, as there often is, because these melanomas shrink over several years, then theoretically it should be possible to biopsy that. There could be a bit more fibrosis, a bit more scar tissue within the tumor. I think after several years there might be lots of scar tissue that might make it more difficult. But after a few months, I think there isn't all that much scar tissue. What do you think?
I think we don't really know, but it would be very important to see if we could. This is something that is possible, and I know there's one paper out there that has looked at doing the gene expression profile after biopsy, and I think we need to be cautious with that because, as we talked about in our group, DNA and chromosomes are very stable, and you can find DNA years after, hundreds of years, and the structure is very similar to the way it originally was. Whereas RNA, if it gets radiated, there's inflammation. RNA is very unstable and very sensitive to changes. So I'm not sure if the class 1, class 2 test would still be valid after radiation. So I think that we may need to rely on chromosomal or DNA-based tests if we're going to pursue that. But I think that's something that I'm certainly very interested in following up on that and perhaps offering a biopsy after the tumor's been treated in people and seeing if we can get some information. In eyes that have been enucleated, we have been able to, on histology, we still see monosomy 3, and that's something that Bertil has commented on, but we also see that. So I think we can still get information from a treated tumor, even though our general thinking is that once it's radiated, there's no more information. There may still be a possibility to get information about prognosis.
Any questions?
Dr. McCannell, or any one of you, the chromosome 3 that is detected in the eye when you're doing the pretest on the tumors, is that a prevalent gene flaw that you'll find in the metastasis in the livers as well, if it's tested?
Yes, actually, that's a good question. Do we see the missing chromosome 3 abnormality in the liver? We certainly, in the liver samples that we have accessed and looked at the cytogenetic changes, we often see monosomy 3 or another form, it's called isodisomy 3, where we see 2 copies of the abnormal chromosome 3. So yes, we do see the abnormality in chromosome 3 in the liver. I don't know, Bertil, if you have experience.
I think there have been some exceptions, but they're very rare. We've also been looking at BAP1, and we don't always see BAP1 in metastases. But there's other lethal mutations, not just BAP1 as well.
So after 10 years fighting metastases, etc., one thing is sure, that not all the doctors collaborate, communicate, and which understand, and it is changing. The last years, things have been changing. But wouldn't it be nice for an orphan disease like ours to have a huge database that can be accessed by whoever wants it, so that researchers would have more info to work with, or doctors to find, I don't know, I'm thinking like from the starting point, when a patient has the melanoma in the eye and goes to the doctor, everything at that point starts getting in a database. If there is a biopsy, if there is profiling, if there is everything. And so it continues, and everything is accessible by whoever needs it, from universities, from drug companies, but something that, as the samples are not, it's an orphan disease and doctors don't get to see as many patients as they would like, I'm speaking more of metastatic doctors, oncologists, etc. It would be nice to have this big bank of data where they could access, and this way probably the research would advance faster. And also for clinical trials, it would be better if everything was better in this database. So I'm thinking, if this doesn't exist, how can we make it happen? How can it exist?
I'll speak about Europe first, because that's where I've been practicing until a few months ago. And perhaps Tara will speak about North America after. Is that a good idea? I agree with you 100%. With a rare disease like uveal melanoma, we must collaborate. So what we did in Europe in 1992, or 1991, 1992, under the auspices of the European Organization for Research and Treatment of Cancer, EORTC, we set up the European Ophthalmic Oncology Group. And I was in the founding committee as secretary for 6 years and as president for 3 years. And thanks to that group, all the ocular oncologists involved in treatment of uveal melanoma got together every 6 months, and we made friends, and we visited each other's hospitals and organized conferences at each other's center. And in Europe, the ocular oncologists really get on very well together. And all centers have got tissue banks. So what we started in Liverpool was a virtual tissue bank, whereby we had a spreadsheet listing all the tissues and all the clinical details relating to that patient, and we shared that information with all the other scientists so that if anybody wanted any samples, they would get them. They would know what we had available, and they would have them. But you've got to be careful with things like that, because you can get some scientists who take away your precious tissues and their experiment fails. So there are certain conditions, certain precautions you must take not to waste the samples, because they're very small samples very often. And the other important thing, sorry, did you want to say something?
No, I understand
Okay.
your point. I was just thinking, at least the info, like as a patient, when you go somewhere and that's fine. I think my voice is loud enough. When you are, you have already had your gene profiling done in one place, and then you go to another place, and they say, "Oh, but we cannot get it." So it seems that there is no communication and not easily accessible information.
Fine. I think I know what answer you're looking for, and I'll try and give that. The other thing I wanted to say was tissue samples by themselves are useless. The tissue sample is only valuable if it comes with patient consent to share the tissues with other centers, to get follow-up information, things like that. And it's very complicated keeping that tissue consent, because you've got a paper consent in the charts, and the charts get incinerated or lost, you've lost your consent. I used to have somebody working almost full-time archiving all the patients' consents and making sure we never lost any of those consents. And apart from consent, you need to have the clinical data. You need to know how big the tumor was, where it was, how extensive it was, and all that kind of thing. Because without that clinical information, the tissue information is useless. And you need to know about survival, whether the patient's alive or not, years later, not 5 years later, but 10, 15 years later. And there are many patients who get lost to follow up. In England, we had a system whereby we're automatically notified every time a patient passed on.